ওকে, আচ্ছা কি পড়াশোনা করছো?
স্যার, আমাদের রেজাল্ট কবে দিবেন?
ভেরি গুড কোয়েশ্চেন। রেজাল্ট পেয়ে যাবা ইনশাল্লাহ, দ্রুতই পেয়ে যাবা। হ্যাঁ, অনেক খাতা দেখতে একটু সময় বেশি লাগে। আজকে তো 6 তারিখ।
আমাদের টোটাল কুইজ কয়টা হবে স্যার?
টোটাল কুইজ আর 2টা হবে। হ্যাঁ, আমি তোমাদের নেক্সট ক্লাসে কুইজের ইয়ে জানাই দেবো, কারণ তোমাদের নেক্সট কুইজের আগে হচ্ছে 2টার খাতাও দিয়ে দিব আর কি। আই গেস নেক্সট ক্লাসেই দিয়ে দিতে পারবো আর কি, 2টাই। অবশ্য ক্লাস আমি খাতা ক্লাসে দিই না, হ্যাঁ, ও তোমাদের এই ইমেল অ্যাড্রেসে চলে যাবে, বোথ খাতা এবং স্ক্রিপ্ট অ্যান্ড মার্ক ইন্ডিভিজুয়ালি সবার ইমেল অ্যাড্রেসে চলে যাবে। নেক্সট ক্লাস
ফাইনাল কবে?
ফাইনাল, 1 মিনিট। ফাইনালের ডেটটা তোমাদের মানে ইয়েতে দেওয়া আছে, ইউসিসি দেওয়া থাকার কথা, ইউএসআইএসএ আর কি। দেখি কত কবে এক্সাম। কোথায় আছে এটা?
স্যার, ওখানে 4 জানুয়ারি দেয়া।
4 জানুয়ারি, তাহলে 4 জানুয়ারিতে ফাইনাল হচ্ছে, 4 জানুয়ারিতে। হ্যাঁ,
ওকে স্যার।
ফাইনাল নিয়ে অত বেশি টেনশন করো না, তোমাদের এই মিড যেরকম হয়েছে না, এক্স্যাক্টলি সেম হবে। সেম 30 মার্কের এক্সাম, সেম একই রকম সময় পাবা। মিডের সিলেবাস ফাইনালে থাকবে না। এই জন্যই মিড হচ্ছে 30তে নেওয়া, যাতে তোমাদের মানে ফাইনাল আর মিড একই রকম হয় আর কি। আচ্ছা, আসো আমরা একটু পড়াশোনা করি। লেকচার 9 and 10, 10, okay. Okay, so have you studied the lecture 9? So if you have any question from lecture 9, you can ask. I'll give you the answer. পড়াশোনা করতে হবে কিন্তু, সময় বেশি বাকি নেই। আচ্ছা, আমি লেকচার 9টা একটু শেয়ার করতেছি। হ্যাঁ, সো লেকচার 9 থেকে কোশ্চেন থাকলে তোমরা করতে পারো, যদি কোশ্চেন না থাকে তাহলে আমি লেকচার 10 একটু ব্রিফ দিব, যদিও তোমাদের লেকচার 10 পড়ে আসতে বলছিলাম। I don't know you guys have seen the lecture 9 or not, I mean lecture 10 or not. Okay, করে গেল। Lecture 9, I mean, if you have any question from lecture 9, please feel free to ask.
Excuse me, sir.
Yep.
You talked about E. coli and the Vibrio.
Yeah.
As if I remember, like you said, also E. coli causes cholera. Is it true?
Not cholera, diarrhea. I mean cholera-like disease.
Okay. So it's not the symptoms and the signs are almost the same.
Yeah, almost same.
Okay.
They both cause diarrhea. However, there is slight difference in the diarrhea, type of diarrhea. Okay, so the diarrhea caused by Vibrio cholerae is more severe. Okay,
Okay.
and one characteristic is watery diarrhea. I mean, diarrhea caused by cholera is it causes rice water stool. This is an you can say an exclusive characteristics of diarrhea caused by Vibrio cholerae. Well, the diarrhea caused by E. coli can also be deadly, depending on the person's health condition. But the diarrhea caused by Vibrio cholerae is always deadly. And diarrhea is a disease which is actually a severe one. I mean, no matter how healthy a person is, if a person get diarrhea and not treated, they will die. Why people die of diarrhea? ডায়রিয়া আসলে মানুষ মারা যায় কেন?
স্যার ইলেকট্রোলাইটস কমে যায় অনেক বেশি।
Yeah, one cause.
Excuse me, sir.
Yeah.
Can you really describe diarrhea as a disease, or not just basically a symptom or sign of a disease? Is it basically a disease or a symptom of a certain disease, in that a certain disease is causing or causative agent is causing diarrhea?
Okay, you actually raised a good point. Well, if we say very scientifically or in a strict sense, diarrhea is a symptom. It's symptom of a disease, right? I mean, in general, we like if someone is having diarrhea, we can say, okay, he got diarrhea. Okay, he is sick. Okay, in general, sometimes we call it a disease, but actually, it is a symptom which can be caused by the infection with different microbes. Because even like uh, COVID-19, some COVID-19 infected patients also suffer from diarrhea. Not all, but some. So in that sense, it's actually a symptom. Okay.
Okay, sir.
Basically, people die of diarrhea because of dehydration and loss of electrolyte. Dehydration is a severe thing. I mean, if our cell get dehydrated too much, মানে সেল থেকে যদি পানি বের হয়ে যায়, dry হয়ে যায় cell, they stop working. And simultaneously, electrolytes are everything. Remember this one thing. I mean, in our body, lots of things are happening. Okay, lots of metabolic things, lots of signaling things. Lots of things are happening in our body, and all those things are controlled by electrolyte, different ions. So if our body lose water and ions, they stop working. I'll give you a very I'll give an example. Our heart is beating, right? আমাদের heartbeat হচ্ছে না continuously, and because of that beat, blood can circulate throughout our body. So this whole process is controlled by electrolyte. uh, More specifically, it is controlled by calcium. Okay, almost many of the things in our body is controlled by sodium and no, no, not calcium, sorry, potassium. Okay, sodium and potassium, lots of these kind of things. This kind of mechanism, like the heart, is controlled by electrolytes, mainly by potassium and sodium. Okay. Methanobacterium. Okay, you know that.
Excuse me, sir.
Yep.
What do you mean by 16S RNA?
RNA.
Ribosomal RNA.
Where was the 16S RNA? Here. Can you please tell me where was the 16S RNA?
It was in lecture 10, basis of classifying the
Archaea.
archaea,
Okay.
the domain for classification.
Lecture 10 or
Lecture 10.
lecture 8? Okay, lecture 10 also have that, I guess. 16S RNA, actually we have to call it 16S rRNA, ribosomal RNA.
Yes.
Okay. Here, right? 16S, where it is? It should be okay. 16S rRNA, okay, ribosomal RNA. So we know what ribosome is, and we know that ribosome is composed of subunit. Okay, as a whole, ribosome is an active organelle. Ribosome can synthesis amino acid. Okay, মানে অ্যাজ এ হোল তো আমরা রাইবোজোম কি, রাইবোজোম তো আমরা জানি এটা একটা অ্যাজ এ হোল অর্গানেল, বাট রাইবোজোমস আর মেড অফ several subunits, structural subunits. So one of the subunits is 16S. S is a unit, like say for example kilogram. Kilogram is a unit for weight. Meter is a unit for length. So S is a unit to measure the you can how can I say, measure the size of a subunit. Okay. Okay, so this unit was developed by a scientist whose name is Svedberg something. I forget his full name. Svedberg something. So from his name, this S came. S হচ্ছে ওই রাইবোজোমের ইউনিট, রাইবোজোমের সাব ইউনিট গুলার মানে ওটা ধরো সাইজ কিরকম, ওটা একটা ইউনিট আর কি। ওকে, সো 16S means it's a subunit of ribosome. Okay, so that ribosome and okay, 16S ribosome. Okay, and this RNA means RNA. Okay, RNA means RNA, but this small r represent ribosomal RNA. So that means this RNA belongs to the 16S subunit of the ribosome. এটা হচ্ছে রাইবোজোমের যে 16S সাবইউনিটটা সেটার RNA আর কি। ওকে, and this classification, I mean the 3-domain classification, is done by on the basis of 16S ribosomal RNA. Now the question is why? Okay, so in our body, okay, so like we have like a DNA, okay, and ribosome, they have RNA. Ribosome are made of RNA. So the thing is, what are these? These are genetic material, right? Nucleic acid. So okay, some of our nucleic acid, they change very rapidly. Okay, basically we call them variable. Okay, because they change frequently. So the nucleic acid which change rapidly or more frequently, we cannot use those nucleic acid to characterize something, to identify an organism. Okay, so I mean যেই নিউক্লিক অ্যাসিড মানে নিউক্লিক অ্যাসিড সিকোয়েন্সের উপর বেস করে, বেসড অন দ্য নিউক্লিক অ্যাসিড সিকোয়েন্স উই ক্যান আইডেন্টিফাই আ অর্গানিজম। Just human is the organism. We can identify by detecting the nucleic acid sequence. E. coli is an organism. We can identify E. coli by nucleic acid sequence. Okay, identifying nucleic acid sequence, or I mean, or all the organisms. We can identify them by using nucleic acid sequence. Now, in doing so, we don't use that nucleic acid sequence which change rapidly. We choose those nucleic acid sequence which change very slowly. This kind of sequence we usually call them conserved sequence. Conserved. Okay. Conserved sequence. So the conserves, I mean, if you think in the sense of evolution, I mean, in time, the change of conserved sequence is very slow. So that's why we choose conserved sequence to identify uh, organisms. Okay. And the 16S ribosomal RNA, these RNA sequence are very conserved. They change very slowly over time. So that's why this is a good option. I mean, using 16S ribosomal RNA is a very good option to identify an organism. Okay, did you get it?
Yes, sir. I got it.
16S রাইবোজোমাল আরএনএ মানে আমাদের ডিএনএ তে আমাদের নিউক্লিক অ্যাসিডে এরকম কিছু রেজিয়ন থাকে যারা খুব ভেরিয়েবল আর কি দ্রুত চেঞ্জ হয়, কিছু রেজিয়ন থাকে তারা খুব স্লোলি চেঞ্জ হয় ইভলিউশনারি। তো আমরা না ওই ওই অংশগুলোকে মানে যেই আমাদের নিউক্লিক অ্যাসিডের যে অংশগুলো খুব দ্রুত চেঞ্জ হয় সেগুলোকে না আমরা অর্গানিজমকে আইডেন্টিফাই করার জন্য ইউজ করতে পারি না, কারণ এগুলো খুব দ্রুত চেঞ্জ হয়। ইভেন দেখা যায় হিউম্যান টু হিউম্যান চেঞ্জ হয়ে যেতে পারে, ঠিক আছে, মানে পার্সন টু পার্সনও চেঞ্জ হতে পারে। সো আমাদের আবার নিউক্লিক অ্যাসিডে কিছু অংশ আছে যেগুলো খুব স্লোলি চেঞ্জ হয়, খুব স্লোলি। ওকে, সো দে মে টেক থাউজেন্ডস অর মিলিয়নস অফ ইয়ার্স টু চেঞ্জ। তো আমরা ওই ধরনের রেজিয়ন গুলোকে নিউক্লিক অ্যাসিডের ওই নিউক্লিক অ্যাসিড গুলোকে ইউজ করি অর্গানিজমকে আইডেন্টিফাই করার জন্য, যে ওকে ঠিক আছে এটা হচ্ছে ইকোলাই, এটা হচ্ছে ধরো স্পাইরোসিট, ওকে এটা হচ্ছে ভিব্রিও কলিডি, এটা হিউম্যান, এটা মাঙ্কি, এই ধরনের। এই জিনিসটাকে না ফাইলোজেনি বলা হয়। হ্যাঁ, ফাইলোজেনি। Basically, I mean, whenever it's phylogeny, it means the similarities and dissimilarities in the nucleic acid among different organism. Okay. Okay, I guess you guys don't have any question from lecture 9 and from lecture 10. Do anyone have any question? Okay, if you don't have any question, I'll try to just give a brief overview. Okay. So, archaea. You know, for a long time, people used to believe that archaea are bacteria. But eventually, scientists realized that they are almost like bacteria, but they have some exclusive characters and they should be regarded separately than the bacteria. তাদের কিছু এক্সক্লুসিভ ক্যারেক্টারিস্টিক্স আছে, এবং হোয়াট ইজ দ্যাট এক্সক্লুসিভ ক্যারেক্টারিস্টিক্স? দে ক্যান টলারেট extreme environmental conditions, various extreme different type of various extreme environmental conditions, like some can tolerate extreme high temperature, some can tolerate extreme pressure, very cold temperature, very high salinity, মানে অনেক বেশি লবণাক্ততা, okay, very high sugar content, okay, high pressure, or like say presence of high toxic chemical, like presence of high sulfur. Okay, so these kind of conditions are not okay for normal microorganisms. Normal microorganisms, even like normal organism, they cannot tolerate this kind of extreme temperature, extreme environmental conditions. But archaea, they can. Okay, They have this spatial capability. So there are different type of archaea. It's not like that the same archaea can tolerate all the extreme environment. No, there are different kind of archaea, and different archaea, they have different uh, how can I say, they have different uh, like different tolerance level, or I mean different bacteria can different type of extreme environment. Like some archaea, they can tolerate extreme high temperature, some archaea they can tolerate very high pressure, like that. Okay, so mainly because of that, they fall into different domain. Okay, so which was basically finally successfully characterized by Mr. Woos. Okay, in his three-domain classification system, he characterized archaea as a different domain. Okay. Okay. And what are those? Okay. However, I mean the archaea, they can be further divided into three kingdom, two major and one minor kingdom. Major kingdom are Euarchaeota, Grenarchaeota. Minor kingdom is Corarchaeota. And how? I mean, not only the three domain, also these archaea are further divided into these three kingdoms based on the sequence of 16S ribosomal RNA. Okay, there are many different type of RNA. Okay, in our body, messenger RNA, transfer RNA, ribosomal RNA. Okay, so usually if we want to specify RNA, I mean, it's better to use this kind of a small letter in front of the RNA to say, I mean, what kind of RNA is this? Okay. Usually, especially for RNA, we usually do this. I mean, we specify, I mean, what kind of RNA is this? Okay. Egglet to huh? If you read this, you will understand these are historical things, simpler things, general characteristics. They are prokaryote. So definitely they are nucleic acid, I mean, hereditary molecule or genetic material will be, hereditary molecule will be DNA. Okay. Actually, all the cells, prokaryote is a cells, right? So all the cells, their genetic material is DNA. There are only some virus whose genetic material is RNA. Okay. Guys, COVID-19 is a RNA virus or DNA virus? What kind of virus it is?
Sir, RNA.
Yeah, it's RNA virus. So except virus, okay, not all the virus are RNA virus. Huh? Some virus are RNA virus. So except the virus, all the other, I mean, okay, virus is actually not an organism, right? We say it, we call it a microorganism, but it's actually not an organism. Anyway, so except the virus, all the other organisms, all the cells there, they have DNA as their genetic material. Okay, anyway, archaea, they have 17S ribosome. Okay, and methanogenesis is the exclusive ability of archaea. I mean, they can do methanogenesis. That means they can produce methane. Methane is a gas. They cannot do photosynthesis. That means they cannot produce their food by their own. So for living or generating energy, they need organic. They have to rely on other things. Okay, organic or inorganic things, because they cannot generate their own food. There are different type of archaea, okay, who who have different exclusive characteristics, like, for example, chemolithotrophs. Okay, chemolithotrophs, they can oxidize inorganic compounds as their source of energy. So if we want to say more easily, chemolithotrophs, they need, I mean, as food, they use inorganic compounds. Okay. For living, every cell, they need to generate energy. Okay. So, for example, we human, can we generate energy from inorganic compound? What do you think? Can we live on inorganic compound? তোমাদের কি মনে হয় আমরা কি ইনঅর্গানিক কম্পাউন্ড দিয়ে থেকে এনার্জি জেনারেট করতে পারি আমরা মানুষরা? এই যা ধারণা তাই বলো না। I mean, it's not necessary that your answer have to be correct, right?
স্যার মনে তো হয় না।
মনে হয় না?
না।
Yeah, you're right. We need organic compound to generate energy. Okay. So every living, look, I mean, it's good to guess, right? And there is nothing wrong in giving wrong answer. It's absolutely fine. Okay, so like, I mean, for to live, every organism, they need to generate energy. Every organism, every cell. If we say more specifically, every cell need to generate energy. Without energy, no cell can survive. Because to survive, the cells need to do various kind of work. Okay, so anyway, so these chemolithotrophs, these archaea, that means these cells, they can use inorganic compounds to generate energy. Okay, and how they generate the energy from these inorganic compounds? By oxidizing them. Okay, by oxidizing the inorganic compounds, they generate the energy. Okay, so for example, sulfolobus. Okay, this is an example of chemolithotrophs, and it can use sulfur, okay, to generate energy. Okay, and this is actually just a chemistry thing, is electron donor. I mean, sulfolobus, they need to oxidize the sulfur. Basically, what happens during oxidation and reduction reaction? This is chemistry. This is all chemistry. Okay, oxidation. What happens in oxidation reduction reaction? Someone have to, I mean, accept the electron which the oxygen provide. Okay, say sulfur, they can accept the electron from the oxygen. That's why they can get oxidized. Okay, and in this chemical process, energy is generated. Okay, that's simple. Okay, so this is just the chemistry, is a electron donor. But as a microbiologist, what we should keep in our mind, that's a sulfolobus, it's a, I mean, this archaea, it's a chemolithotroph. It can use sulfur as their energy source. Okay, and how they generate energy by sulfur, from sulfur? By oxidizing the sulfur. Okay, okay, chemoautotrophs, they can use only carbon dioxide as their source of carbon. Okay, they can use carbon dioxide as their only source of carbon. Source of carbon can be different things, like say for example glucose, it has also carbon. Okay, theoretically you can generate carbon from like sugar or various carbohydrates. Okay, all the carbohydrates are carbon. Okay, however, the chemoautotrophs, they can use only carbon dioxide as their carbon source. Example, pyrococcus, chemoorganotroph can oxidize organic compounds as their source of energy. That means chemoorganotroph, they use organic compounds as their food, source of energy. Methanogen, they produce methane from organic substrates, methanotropes. They use methane as their source of energy. Okay. Picrophiles, this kind of archaea, they can, okay, they need extreme low pH as a growth requirement. Extreme low pH, that means very acidic pH. Okay, so these were some different archaea. Okay, and this slide, it's actually like it's a summary. You can say it's a summary slide of the extreme environmental habitat of archaea. Okay, I mean, this kind of physiological condition, or more generally say, the different extreme environments where the archaea survive. But not only survive, I mean, actually, this kind of extreme environments are required for those respective archaea. Say, hypersaline environment, 4 molar sodium chloride concentration, extremely high, right? 4 molar sodium chloride concentration, extremely high salt concentration. Anoxic sediment in the bottom of water body without molecular oxygen in the, it will be anoxic condition. Anoxic means without oxygen. So in the bottom of ocean. Okay, why actually? Well, I mean, okay, we actually know very little about what happens in the bottom of deep ocean. Okay, but anyway, so like say for example, the organism which usual organism, if we say the usual organism, they actually need oxygen, okay, so to survive. But in the bottom of like ocean, or in the sediment of water body, those sediment are anoxic. Okay, there is no oxygen there. Okay, so the normal organism cannot survive there. But archaea are not normal, they are abnormal. Okay, it's just a, I mean, I was just joking. They are abnormal in a sense that they don't live in the normal environment. They're special. Okay, they're not abnormal, they're special. Okay, then submarine habitat, deep ends of the ocean. In the deep ends of the ocean, the pressure is extremely high. Also, sunlight is low, oxygen is low. So, and in an archaea can survive in this kind of extreme condition. Thermal vent, the temperature of thermal vent is very high. Okay, so like black smoker is a thermal vent which emits heavy concentration of sulfur and other volcanic minerals. Okay, যে ইয়া থাকে না, আগ্নেয়গিরি থাকে না, অনেক যেগুলো হচ্ছে তোমার একটু মৃত প্রায় অলমোস্ট ডেড আগ্নেয়গিরি, volcano, they frequently emit smoke, black smoke, and those smoke are heavy with sulfur. Well, do you know no volcano is regarded as dead nowadays? When I was a kid, I mean, I was in the school, our textbook used to say that there are like dead volcanoes and live volcanoes, but this notion has changed. Okay, so the archaeologist or I mean the puppet, I mean the scientist who deal with this kind of things later, I mean so maybe few years back they announced that no volcano is dead. Okay, any volcano can erupt anytime. Anyway, so these are example of some of those extreme environment where different archaea live. Okay, then please read carefully the following lectures. I mean, like so in the following slides. I mean, what are there? Like, say for example, now we know that archaea can survive in different extreme environmental condition, but how? Okay, they should have some speciality. I mean, how they can survive. Why the normal cell cannot survive in the extreme environment, but why the archaea can survive? There must be something in the archaea. Actually, it is true. Okay, indeed, the archaea have some speciality. Okay, they have some special mechanism by which they tolerate this extreme environment. Okay, so in the following lecture slides, you will find those kind of things. So, for example, extreme halophiles. Okay, extreme halophiles means they are representing a group of archaea. Extreme halophiles. Halophiles means, okay, so halophiles means they love high concentration of salt. মানে ওই সল্টের কনসেন্ট্রেশন বেশি থাকলে মানে হ্যালোফাইলস যারা ফাইলস মানে কি ফাইলস মিন্স দে লাভ ওকে হ্যালোফাইলস দে লাইভ হ্যালো সো দিস মিন্স দে লাভ high concentration of salt, and extreme halophiles mean they love extreme concentration of salt. Okay, say an extreme halophile needs 1.5 molar sodium chloride for growth. Okay, even some halophiles, some extreme halophile, they can even survive at 5.5 molar sodium chloride. Okay, concentration. This is extremely high. Okay, so this amount is found in Dead Sea. You know you all heard the name of Dead Sea, I guess. Okay, so the salt concentration of Dead Sea is quite high. No, not quite high, extreme high, 5.5 is very high, right? Okay, so the thing is, normal my organism, say, okay, microorganism, they cannot survive in here in this kind of high concentration. Why the halophiles can? Okay, so let's see why. Okay, so first is why the halophiles need high concentration. Why? I mean, they need why the halophiles need high salt concentration? So the glycoproteins and cytoplasmic proteins of halophiles need high concentration of cations to stabilize. Okay, and these proteins have a high amount of acidic amino acid residues which can only stabilize in the ionic solutions. Okay, so like glycoproteins and cytoplasmic proteins. Okay, so these proteins made the structure of the cell, structure of the archaea. Archaea is a cell, right? So glycoproteins and cytoplasmic proteins. Okay, for a cell to survive, they need these proteins. মানে আমাদের যে সেল থাকে, সেলের সার্ভাইভ করতে গেলে গ্লাইকোপ্রোটিনস এবং যে বিভিন্ন ধরনের সাইটোপ্লাজমিক প্রোটিন, মানে যে প্রোটিনগুলো সাইটোপ্লাজমে থাকে, ওকে, দ্য গ্লাইকোপ্রোটিন। সো এই প্রোটিনগুলো দরকার, এই প্রোটিন ছাড়া তো কোনো সেল সার্ভাইভ করতে পারবে না। ওকে, because for the various mechanisms or metabolic or anabolic things that the cell need to do to survive, okay, they require this kind of protein. Okay, so in case of halophile, these proteins are only stable when the concentration of this salt is high. Okay, so at low concentration of salt, these proteins of halophiles are not stable. Because if the proteins are not stable, how they will work, right? So this is the reason why halophiles need high concentration. Okay, because at high concentration of salt, cations, especially sodium and potassium, the glycoproteins and cytoplasmic proteins of halophiles are stable, so they can perform properly. Otherwise, they can. Okay, so this is why they need high concentration of salt. Now, another question that then, okay, we understand the stability of this protein, glycoproteins or cytoplasmic proteins. Okay, we understand that, but whenever we put a cell in high concentration of salt, usually this cell they can get burst out. এখন ফেটে যাবে যখন কোনো সেলকে যদি আমরা হাই কনসেন্ট্রেশন অফ সল্টে রাখি, ফেটে যাওয়ার কথা আর কি। আর্কিয়া কেন ফাটে না? Why the archaea they don't get disrupted? Okay, lysed. Lysed মানে ভেঙে যাওয়া. Okay, lysed means lysis means to get destroyed. Okay, ভেঙে যাওয়া. Lysis মানে. Okay, so why? Now, let's see what happened for normal organism. So when organisms adapted to 0.8% sodium chloride solution, this is means actually this represent the usual organisms, like say our human cell. They are adapted to 0.85% sodium chloride solution. Okay, like except the archaea. Okay, so all the other organisms, they are adapted to this sodium 0.85% sodium chloride solution. This is the normal sodium salt concentration. Okay, so when these organisms, they are in extreme they are in high salt concentration. Okay, so what happened, they have to face extreme osmotic pressure. So what does it mean? I mean, organism adapted to 0.85% sodium chloride means basically actually the salt concentration inside the cell are like this, around 0.85%. Okay, so if these organisms, these cells are put in, if we keep these organisms in high salt concentration, what will happen? There will be a extreme osmotic pressure, because the salt concentration inside the cell is low, the salt concentration outside the cell is very high. Okay, so because of this difference in concentration, the osmotic pressure will be very high, extreme. So the cell will get disrupted, right? Because because of the extreme osmotic pressure, all the water content of the cell, they will get out of the cell. That means they will get disrupted, they will get lysed, because when all the water will get out of the cell, they will get dehydrated and they will die. I mean, you can say they will be disrupted or destroyed or lysed, whatever. They will die. Okay, then I mean, this is the usual things that should happen, but why the archaea don't die? So that means that actually the archaea, they should have the capability to withstand this extreme osmotic pressure. They should have the capacity to tolerate this extreme osmotic pressure. এই যে এক্সট্রিম অসমোটিক প্রেশারটা থাকে, এটা আর্কিয়াদের এটা টলারেট করতে হবে সামহাও। যদি টলারেট করতে না পারে তাহলে তো তারা মরে যাবে। ঠিক আছে? Okay, so now how they do that? The archaea. Okay, so the halophiles, that means this halophiles, this archaea, the halophiles মিন আর্কিয়া। ওকে, আচ্ছা, এটা কিন্তু আমরা কিন্তু আর্কিয়া মানে সব আর্কিয়া নিয়ে কথা বলতেছি না, আমরা হ্যালোফাইল নিয়ে কথা বলছি। বিভিন্ন ধরনের আর্কিয়া আছে, এক এক ধরনের আর্কিয়ার এক্সক্লুসিভনেস এক এক রকম। সো হ্যালোফাইলস তারা এক ধরনের আর্কিয়া, তাদের স্পেশালিটি কি, তারা হাই সল্ট কনসেন্ট্রেশন টলারেট করতে পারে। এনিওয়ে, সো দ্য হ্যালোফাইলস, দে ক্যান stop water diffusion through maintaining a positive pressure inside the cell. Okay, this can happen either by producing glycerol phosphatase or accumulating potassium ions. Okay, so why the normal cell die when they are in high salt concentration? Because of the concentration difference between inside the cell and outside the cell. The osmotic pressure is high, so that's why the water, they get diffused out of the cell. That means the water, they get out of the cell. সেলের ভিতর থেকে পানি বের হয়ে যায়। ঠিক আছে? So if the cell want to survive, they need to stop this. I mean, if the cell, they can stop the diffusion of water, then they will survive in high salt concentration. যদি সেলের মানে কোনো সেল যদি এখন তার ভেতর থেকে এই যে অসমোটিক প্রেসারের জন্য পানি বের হয়ে যাচ্ছে, এটা যদি কোনো সেল ঠেকাতে পারে, তাহলে কিন্তু তারা বেঁচে থাকবে। ঠিক আছে? হ্যালোফাইলস ক্যান ডু দ্যাট। Okay, so theoretically, the halophiles, when they are in the high salt concentration, the water content should get out from the cells, right? The what? Okay, but the halophiles, they can prevent it. That means even they are in the high salt concentration, they can prevent the getting out of the water. So how they can do that? They do it by this way. Archaea can produce some halophiles. Sorry, some halophiles, they can produce glycerol phosphates. Glycerol phosphates is a thick chemical. Okay, so if the cells, they produce glycerol phosphate, that means the concentration inside the cell will be high. So it will balance the pressure difference. So osmotic pressure, there will be no osmotic pressure, so water will not, diffusion will not occur. এই ডিফিউশনের বাংলা জানি কি কারো মনে আছে? এই জানো তো, এটা পড়ছো না? এগুলো তো ছিল তো 9-10 না?
ব্যাপন।
ব্যাপন, ব্যাপন না? কি বলে?
Yes sir, ব্যাপন।
ব্যাপন, ওকে, ব্যাপন, ব্যাপন, হ্যাঁ, ভুলে গেছিলাম। হ্যাঁ, আমি নিজে বাংলা মিডিয়ামের স্টুডেন্ট, আমি এগুলা জানতাম, কিন্তু অনেক আগে পড়ছি তো এই জন্য মনে নেই আর কি। আচ্ছা, যাই হোক, সো যে ব্যাপন আর কি, হ্যাঁ। সো I mean, as some halophile, they can produce glycerol phosphates, so that by this they can increase the concentration inside the cell. So it balances the like the high concentration which is outside the cell. Okay. Another thing, some halophiles, they can accumulate potassium ion inside the cell. Okay, so some halophiles, they can produce glycerol phosphate, some can accumulate potassium ions, or some can do both. Okay, some halophiles, they can do both. They can produce glycerol phosphatase. It's a thick chemical, and at the same time, some can accumulate potassium ion. By doing these things, they increase the concentration inside the cell. So by this way, they can survive in a place where the concentration is high. Okay. মানে এইসব জিনিস প্রডিউস করতে পারলে, মানে সমস্যাটা কি? সমস্যাটা হয় আসলে, মানে কেন নর্মাল অর্গানিজম বেঁচে থাকে না হাই কনসেন্ট্রেশনে? কারণ হচ্ছে সেলের ভিতরে বাইরের কনসেন্ট্রেশন ডিফারেন্ট থাকে, ডিফিউশন ঘটে আর কি, অসমোটিক প্রেসারের কারণে ওয়াটারের ডিফিউশন ঘটে। সো এটা ঠেকাতে পারলেই বেঁচে থাকতে পারবে। সো হ্যালোফাইল যারা, তারা এটা এখন এটা ঠেকানোর উপায় কি? অ্যাকচুয়ালি এটা ঠেকানোর উপায় হচ্ছে আইদার তোমার সেল ওয়াল করে ফেলতে হবে খুব মজবুত, যে সেল ওয়াল হচ্ছে এতখানি, আই মিন দ্য সেল ওয়াল ইজ সো স্ট্রং দ্যাট ইট ক্যান prevent the osmotic pressure. So cell wall ke jodi eto if you can make the cell wall that strong, it can do that, but it's actually very difficult. Okay, the osmotic pressure is so high that you cannot make the cell wall that strong. Well, the cell wall of halophiles is stronger. Okay, stronger than usual microbes, but it's not it's not possible for this kind of cells to make the cell wall that strong to prevent the osmotic pressure. So what option they have? They can increase the concentration inside the cell. যেহেতু সেল ওয়ালকে অতখানি শক্ত করা সম্ভব না, সো অপশন কি থাকে? অপশন থাকে হচ্ছে সেলের ভিতরে আমরা কনসেন্ট্রেশন বাড়াই দিব। যদি সেলের ভিতরে কনসেন্ট্রেশন বাড়াই দিতে পারি, তাহলে সেলের ভিতরে বাইরের কনসেন্ট্রেশন সেম হবে, আর অসমোটিক প্রেসার কাজ করবে না আর কি, হ্যাঁ। সো halophiles, they do that. They increase the concentration inside the cell. How? By producing glycerol phosphatase, this thick chemical, or accumulating potassium ion inside the cell. So example হচ্ছে, these are some example of extreme halophiles. Halobacterium, halo কি হবে এটা, রাবরাম, okay, haloferax, হ্যাঁ, হ্যালো আরকুলা, হ্যাঁ, কিছু একটা হবে, হ্যাঁ। I think I'm pronouncing in a wrong way of most of these organisms. It Google থেকে exact pronunciation দেখে নিন। Anyway, so methane is simple. How the some archaea, they produce methane. Okay, so you will understand if you read this. And if you don't understand, just ask me question in the next class. Okay. So anyway, so this is why the halophiles need high concentration, and this is how they tolerate the or survive in the high concentration of salt. So like in this slide, for thermophiles, okay. So thermophiles, from the name we can guess, right? Thermophiles, they need high temperature. Okay. So most of them are obligate anaerobic chemoautotroph. Obligate anaerobic. I mean, they cannot survive in the presence of oxygen. They need anaerobic condition without oxygen condition. And they grow optimally at 55 degrees Celsius. But even there are some extremophiles which can grow at 100 degrees Celsius or above 100 degrees Celsius. Anyway, so okay. So here the mechanism why the thermophiles, I mean how the thermophiles they survive in high temperature. And this slide you will find why the acidophiles they survive in high temperature. Okay. So when you read this, please try to understand this way. I mean the way I tried to describe this one. Okay. I mean when you will study, please ask question to yourself. Studying is not like just memorizing the thing. You have to ask question to yourself. Like I frequently say, ask me question. But when you will be able to ask question, when you will ask the question to yourself first, right? এই পড়ার সময় মানে আমরা যে বলি বুঝে বুঝে পড়া মানে কিন্তু এটা যে কি হচ্ছে, কেন হচ্ছে, কিভাবে হচ্ছে, সায়েন্সে এই ধরনের কোয়েশ্চেন গুলো জিজ্ঞেস করতে হবে এবং এইভাবে কিন্তু বুঝে বুঝে পড়তে হবে। হ্যাঁ, অনেক কিছু ভুলে যাবে, ইটস ফাইন, ওকে, বাট তুমি যদি একবার বুঝে বুঝে পড়ো তাহলে ফিউচারে দেখবা যখন একই জিনিস তুমি আবার পড়বা তখন খুব দ্রুত এটা মানে মনে চলে আসে আর কি। হিউম্যান ব্রেইন ইজ অ্যামেজিং থিং। When we study science, we should basically there are three questions we should ask frequently: what, why, how. What is happening, কি হচ্ছে, why it is happening, কেন হচ্ছে, and how it is happening, কিভাবে হচ্ছে। So we have to frequently ask this question: what, why, how. ঠিক আছে? These are not unlike microbiology, biotechnology, biochemistry. So this kind of subject need passion. You have to love science. Okay, you have to love biology. You need to grow the passion. CG is তো এই বেচেই খাইতে হবে নাকি? শোনো, আমরা হচ্ছে কেন পড়াশোনা করি, কারণ আমাকে খেতে, হ্যাঁ, খাওয়া না লাগলে কি আবার সায়েন্স পড়তাম? আমি পড়তাম, আমিও পড়তাম না, রাইট? পেট আছে তাই পড়াশোনা করতে হয়। বাট দ্য থিং ইজ আমাদের সায়েন্স বেচেই খেতে হবে। সায়েন্স বেচে খেতে গেলে আমাদের সায়েন্স বুঝতে হবে। তুমি যদি নিজেরা ভালো মানে ধরো মাইক্রোবায়োলজিস্ট হিসেবে গড়ে তুলতে পারো, চাকরি বাকরির অভাব নেই দুনিয়াতে। Okay, so what happens? I mean, how they survive at high temperature? Okay, what happened for normal organism like mesophilic organisms? Mesophilic organisms, like say we human, we are mesophilic, we are mesophiles. E. coli, bacteria, it's a mesophiles. Okay, they need normal temperature. So in case of mesophilic organisms, high temperature denatures protein, rupture the membrane and melt the DNA and RNA. That's why the mesophytic organisms, they cannot survive in high temperature. কারণ হাই টেম্পারেচারে তাদের প্রোটিন ডিনেচার হয়ে যায়, মেমব্রেন সেল মেমব্রেন রাপচার হয়ে যায়, ভেঙে যায়, ডিএনএ আরএনএ গলে যায়, নষ্ট হয়ে যায়। ওকে, so that's why they cannot survive in high temperature. But then why the hello guys, sorry, somehow I got disconnected suddenly. There was a network error. Okay,
Okay, sir.
my Wi-Fi was gone, so me too. Anyway, so where I was in the thermophores. Okay, sorry, there is no file in the share, right? I think it's gone. Okay, just give me one more minute. Where the okay, here it is. Okay, here it is. Here it is. Okay, so where I was here. Okay, so why the thermophiles, they can survive in high temperature. Okay, to survive in high temperature, they have to prevent these kind of things. They have to prevent that their protein will not denature, and like the membrane should not get ruptured, the DNA RNA should not get melted down. মানে এখন থার্মোফাইলসরা যদি এই ধরনের মানে হাই টেম্পারেচার সার্ভাইভ করতে চায়, সো তাদের এই জিনিসগুলো ঠেকাতে হবে। কারণ হাই টেম্পারেচারে প্রোটিন ডিনেচার হয়ে যায়, প্রোটিন এত হাই টেম্পারেচার টলারেট করতে পারে না, প্রোটিন ডিনেচার হয়ে যায়, মানে প্রোটিন ভেঙে চুরে যায়, মেমব্রেন ভেঙে চুরে যায়, ডিএনএ আরএনএ গলে যায়, ভেঙে চুরে যায়। ওকে, সো এগুলো ঠেকাতে পারলেই সার্ভাইভ করতে পারবে। ইনডিড thermophiles can do that. Thermophiles can prevent this kind of things. How? Thermophiles have high basic proteins, basic, acidic basic. Okay, basic, that basic. This basic means that basic, acid base, that basic. Okay, so thermophiles have high basic proteins surrounding the DNA, so that the DNA is stable at high temperature. Okay, I mean the DNA of the thermophiles, they have an extra layer of protection. So high basic proteins, they surround the DNA of the thermophiles. So by this, the DNA get stable, that even at high temperature, the DNA do not get destroyed. They don't melt down. কারণ হচ্ছে একটা হাই বেসিক প্রোটিন এই ডিএনএ কে সারাউন্ড করে রাখে, মানে বলা যায় কি একটা আবরণের মতন ঘিরে রাখে আর কি। ওকে, সেই জন্য এই ডিএনএ গুলা হাই টেম্পারেচারও, মানে থার্মোফাইলসের ডিএনএ হাই টেম্পারেচারও স্টেবল থাকে। ওকে, so then the tetraether residue in the cell membrane lipids allow them to tolerate high temperature. I mean the cell membrane of thermophiles, okay, they contain this chemical tetraether. Okay, it's a chemical, right? It's a chemical compound, ether, but tetraether. Okay, so thermophiles, they have this tetraether in their cell membrane. Okay, cell membrane lipids. So this make the cell membrane more stable, allow them to tolerate high temperature. Okay, so this is the underlying secret or mechanism by which thermophiles they can tolerate the high temperature. Okay, and one example is thermoplasma. Okay, just one more things. Acidophiles, they love high acid concentration. I mean, they love acid. That means low pH. Okay. So normal microbes, they cannot survive at low pH because low pH destroys the cell membrane due to destruction of membrane phospholipid. Okay, the membrane of cell contain phospholipid and phospholipid. Okay, lipid. So at low temperature, at low pH, that means acidic pH, this phospholipid they are destroyed. Okay, so that's why usual cells they cannot survive at low pH. However, acidophiles they have unusual amino acids that are sensitive to alkali but stable at low pH and impermeable to hydrogen ion. Okay, I mean the acidophiles. Okay, so they have unusual amino acids in their body and also in the membrane. Okay, so why this amino acid is unusual? Because these amino acids they are stable at low pH. That means they are stable at acid. Okay, so that's why they can survive at low pH because they are stable at they are stable at acidic condition at low pH and also impermeable to hydrogen ion. Okay, and the cell membrane of the acidophiles they are like that. They prevent the movement of hydrogen ion. Okay. Okay. At normal pH, the acidophiles they cannot survive because at normal pH, the cell membrane of acidophiles disintegrate. নর্মাল পিএইচে এই অ্যাসিডোফাইলসের সেল মেমব্রেনটা ডিসইন্টিগ্রেট হয়ে যায়, ভেঙে চুরে যায় আর কি। ওকে, and that kill the acidophils. Okay, so they cannot survive at normal pH. Okay, so this slide, please study this slide, and I mean the next slides, please study these things. Okay, these are not that difficult. So like archaea and evolution, these are interesting thing. Archaea have some similarities with eukaryotes. They have some similarities bacteria. Okay, and this is already I have discussed. I mean, why the archaea they can, I mean, how the archaea survive in very different extreme environment. Okay. Okay, so please study these things and ask me question in the next class. Okay, one last thing, I actually forget to mention a very important thing about archaea. I mean, why archaea are called extremophiles? So why we need to study archaea? Okay, archaea live in the very high temperature, very high salt concentration. Who cares? Okay, okay, they live in the high volcano. So then what? হ্যাঁ, মানে হাই তারা হচ্ছে কি এক্সট্রিম এনভায়রনমেন্টে বসবাস করে, তো আমার কি? ওকে, আমার কি কারণ আছে? I mean, why we need to study archaea? Because they are extremophiles and they have industrial and research importance. Okay, it is thought and already that these archaea they have a special chemicals, like they have a special kind of enzymes, they have a special kind of chemicals which may have industrial importance. Like one very good example is and also like if we so I'll give you the answer. Okay, give you the example before that. Like they can tolerate extreme environment, so if we can find out the mechanism, we can apply this knowledge in genetic engineering to modify the organism in that way, usual organism, so we can use them more efficiently in the industry. Okay, another example, okay, very good example of the chemicals that archaea produce. So there is an enzyme named polymerase enzyme. Okay, just now please remember this thing. Okay, polymerase enzyme is necessary for in molecular biology. Okay. So I'll give you one more specific enzyme. So it's just only one use. We all know the RT-PCR, know about the RT-PCR detection, RT-PCR of COVID-19, right? We all heard that the COVID-19, they are detected by PCR. Okay, technique or method. Okay, there are other ways to detecting COVID-19, which are nowadays, I mean, the people are starting using them, but the gold standard method, which is used currently to detect the COVID-19, is by RT-PCR. And for this method, we need a enzyme. An enzyme, its name is polymerase. Okay. So the polymerase enzyme, okay, this is only a small use of polymerase enzyme. The whole molecular biology, okay, in the whole molecular biology, polymerase enzyme is the most important thing. Okay. And the first polymerase enzyme was identified or discovered from archaea. Okay. Stereothermophilus. Okay, this is a one archaea, and the polymerase enzyme was first discovered from a archaea. And a revolution happened in the molecular biology area then. Okay. After polymerase enzyme was discovered from archaea. Okay. Okay. Nowadays we don't use archaea to produce polymerase enzyme. We use basically, actually, we can use E. coli to produce the archaea by using genetic engineering. But it was first discovered there. Okay. So if we study about archaea, people believe that, and actually it should be, because they can survive in extreme environment. That means they have a special things there in them. They produce special chemicals. They have a special mechanism. So if we can discover those mechanisms, if we can discover those chemicals, we can use them. Right? We can use them in the industry, in the research. That means for the betterment of the human and nature. Okay. So extremophiles are important. Unfortunately, very few microbiologists, they are not only microbiologists, but biologists, they are, I don't know, they are interested in extremophile. Most of the biologists, they are interested in human, I mean, the medical related things, but extremophiles is a good area to study in the future. Okay. Okay. Anyway, that's all for today. Please study lecture 10 and also please study lecture 11. Okay. Please do that. Otherwise, we cannot finish. We will not be able to finish in time. ভাই একটু পড়াশোনা করতে হবে ভাই জানারা, হ্যাঁ একটু পড়াশোনা না করলে তো হবে না। সো লেকচার 11 একটু পড়ে আসেন, 11 12 পারলে একটু পড়ে আসেন, হ্যাঁ এগুলা পড়লে হচ্ছে উপকার হবে, ক্ষতি হবে না। আচ্ছা 11 12 লাগবে না, শুধু 11 পড়ে আসলেই হবে। Please study lecture 11 and ask me question in the next class.
স্যার আমরা মিড টার্ম আর কুইজের রেজাল্ট গুলো কবে পাবো?
নেক্সট ক্লাসে। নেক্সট ক্লাসে একটু আমাকে মনে করে দিও তো, সেকেন্ড কুইজের ডেট দিতে হবে। হ্যাঁ
ঠিক আছে
স্যার।
ইয়েস স্যার।
স্যার আমাদের কোনো অ্যাসাইনমেন্ট নাই?
ভেরি গুড কোয়েশ্চেন। হ্যাঁ অ্যাসাইনমেন্টও দিয়ে দিব, দাঁড়াও লিখে রাখি, ভুলে গেছিলাম। আচ্ছা এই যে লিখে রাখছি। হ্যাঁ
স্যার।
হ্যাঁ জি স্যার।
অ্যাসাইনমেন্ট কয়টা আছে স্যার?
আচ্ছা অ্যাসাইনমেন্ট যেটা হচ্ছে, আমি তোমাদেরকে একটা অ্যাসাইনমেন্ট দিব আর কি, ওখানে দুইটা কোশ্চেন থাকবে, দুইটা কোশ্চেনের আনসার দিয়ে দিলেই হবে। মানে বলতে পারো যে ওই দুইটা অ্যাসাইনমেন্ট কিন্তু একটা কোশ্চেনই থাকবে। ওকে। And oh sorry Kevin, yeah, I mean for assignment basically you will have, okay, you will have two assignment, but it will be like I will give you one assignment question. There will be two question, so you will just answer that question, and that deadline will be extended. Okay, so don't worry about that. I will give you a more than enough extended deadline to submit the assignment. Okay. And I have already written down that I have to find the assignment topics. In the next class, please someone remind me. Okay. Okay,
sir.
guys. Then see you in the next class. Till then, be safe. Bye bye.
আসসালামু আলাইকুম
Assalamualaikum.
Thank you,
Assalamualaikum.
sir.
আসসালামু আলাইকুম স্যার